to welcome Dr. Mohammad Haskali from Peter MacCallum, Chief Radiopharmaceutical Science, to present his work on a radiopharmaceutical company in a test tube.
Thank you, Joanne. Well, hello again, everyone. Today I've got my research hat on, so thank you so much for the opportunity to present here again. We all know that drug delivery, conventional drug delivery, is a time-consuming, laborious, and a very expensive task. So how can we learn from the biological system so that we can actually improve on the drug discovery capacity? How can we develop a pipeline without needing to spend all those years and all those kind of expertise and time to make the development of next-generation theranostics feasible in a timely manner? Before I go to the next slide, I would like to emphasize why we're looking at the discovery of novel theranostics. You might have already heard moving beyond PSMA, but I actually think we need to move beyond conventional wisdom, which means in this space and arena, I don't think the keys to some of these questions lie in targeting some of the conventional receptors like bombesin or the FAPs. I think we need a paradigm shift in this domain to enable some transformative work to happen in this space. Of course, easier said than done. So first of all, you all know what theranostics are. I'm not going to bore you with this again. I think it's been explained several times. But I will say, though, that this image, which you see on Google, is a gross generalization, really, of what should look like this, right? Theranostics is a very complex field. It is not as simple as tethering something through a linker to a chelator to a radionuclide, okay? So where the carrier or the targeting agent could be an aptamer, could be a peptide, could be a small molecule, it could be a protein, it could be an antibody. And the linker could be of all sorts. And then the payload could be different types of radionuclides. So theranostics is a subset of a general type of therapies known as targeted therapies. So here I would like to pause a bit. So when we're talking about the discovery of next-generation theranostics, what are the key criteria? Unfortunately, if you go and try to read literature reports, you won't actually find the answer to this. So you pretty much have to develop your own standards. So what is a good target? What constitutes a good target for a theranostic development? First of all, I think we need to move away from the conventional mindset. That is, it needs to be pharmacologically potent and active on the target. We all know that we are injecting sub-pharmacological doses of radiopharmaceuticals, right? When we're staying, just to stay true to the tracer concept. So the tracer concept means you are administering sub-pharmacological doses. So what it does, how it acts on the receptor, is less important, though I'll be very quick to add that it's not completely irrelevant. It's a bit like the yin-yang sign of the Taoism. There is a white dot in the black circle, and a black dot in the white circle. If you think it's a straightforward thing, then you probably haven't understood the complexity of this matter. And however, the two most important factors that revolve around a successful target for theranostic application are actually the high density. So it needs to be very high density. I'm not talking about just mere overexpression of 1 or 2 or 10 folds. I'm talking about 100 to 1,000-fold overexpression. Okay, and the second parameter that governs a successful theranostic against a novel target is the high affinity. In fact, the binding potential of a radiopharmaceutical is mathematically characterized as Bmax over Kd. That is the receptor density over the affinity of the theranostic. These two parameters are the two most important parameters when you're looking at a theranostic agent. And so the target, of course, also has to be a cell membrane expression. It cannot be intracellular. It has to be extracellular, though some might think that this is probably a dogma. I don't know, but up until now, all the solid examples we have are for extracellular domains. And that extracellular domain has to be properly folded, right, to enable the binding of agents with high affinity. And ideally, you would be able to validate this theranostic target before you even commence the theranostic development process, which is really not the case. Theranostic development historically has actually started quite late in the development process, as you might see here, or in the following slides. So We have a large range of protein targets that we can utilize for theranostic development, including enzymes, membrane carriers, ion channels, and receptors. What we've really targeted up until now is merely receptors and enzymes. Receptors, for example, for somatostatin 2 receptors, or enzymes such as PSMA and FAPS. So there is still a broad range of proteins that we really haven't targeted, and it's questionable whether they can be utilized for the effective targeting of theranostics. I think there is good grounds there for some basic science research to occur just to illustrate the possibility or just to investigate some of the possibilities that lie ahead in this space. Now, the conventional drug discovery starts with a discovery of a hit molecule, which is the identification of a compound with the desired activity against the target. Then you would optimize the hit into a lead, okay, which is a limited optimization really to enhance its performance against the hit. And then eventually you generate a lead optimization from there to enhance its drug-like properties. Historically, theranostic really commences at that step. So most of the theranostics we have in place are based on some earlier work that have already identified the hit, that have already taken the hit to lead, and you already have a lead, and you basically optimize that through what we call structure-activity relationship studies to tailor it for a radiopharmaceutical. However, this is very limited. Now, you heard all the talks before me. There are a lot of proteins coming that are really, really interesting to target. And so how do we go about improving this process? And this is where this pharmaceutical company test tube concept comes. Okay, so we are employing or integrating mRNA display in our technique. mRNA display, basically what it does is it mimics a biological system. It expresses 10 to the 12 unique cyclic peptides in a test tube. Okay, and then those 10 to the 12 unique cyclic peptides can be stratified over a mobilized target in a single day. And so only those peptides that are bound to the target remain on the target, and everything else gets washed off. And you repeat this cycle several times until you get an enrichment of those peptide binders, which then you can synthesize and validate their binding using surface plasmon resonance. So this technique, I like to call a pharmaceutical company in a test tube, or you can call an effective time machine that shrinks really the drug discovery times from a number of years to a few weeks, when it works, of course. It doesn't work every time. So we've currently established this technique, or we're establishing this technique at Peter Mac, and we've applied it against several targets. But why mRNA display in particular? Because many of you are probably familiar with phage display and other peptide discovery technologies. mRNA display actually allows you to develop some of the largest peptide libraries in vitro. So what that means is it translates into a higher affinity ligands that you can discover through it. And it affords cyclic peptides, which are ideal candidates for theranostic application because they occupy like a Goldilocks zone between antibodies and small molecules whereby they attribute the best characteristics for theranostic application. So applying mRNA display, we've been able to identify some very cool cyclic peptides against novel targets discovered by our biologists, including Luke Furyk, and in collaboration with Professor Hoffman, we've identified some really cool proteins that are overexpressed on PSMA-negative prostate cancer, including the lethal neuroendocrine prostate cancer, amongst many other cancers. And we've employed mRNA display to discover these unique cyclic peptides against them that are excellent candidates for the development of next-generation theranostics. And so we've established a transformative platform at Peter Mac to enable the synthesis of cyclic peptides using this automated module here, which is a microwave-assisted peptide synthesizer, which enables really the synthesis of up to 12 peptides in a single batch, so we can really make hundreds of peptides a week if we wanted to. Of course, we still have to process them, and that's often the rate-limiting step. But these peptides are optimized and tailored for theranostic application. And finally, in my last slide, I would like to say that the next steps from here, so in parallel, we also have to think about assay development and the preclinical models and what sort of models are going to be suitable to investigate those novel theranostics. And how do we go about their clinical translation? And this is where you really need a multidisciplinary team to assist you with. Finally, I want to thank Professor Hoffman, who is a fantastic mentor and an excellent collaborator. Thank you, Michael, for giving me the opportunity in this conference. And my, biologist guru, Associate Professor Luke Furyk, thank you so much for this wonderful collaboration, this opportunity to do some of this great work. And of course, lastly, to my research team here, thank you so much. They do all the work, I do all the talking. So thank you so much.